This video will help you understand how mutations can be detected in two different genetic systems. First, the Ames test uses a selection in bacteria to determine if a compound is a mutagen. Second, Muller performed a screen in a more complex organism, the fruit fly Drosophila, to ask if X-rays cause mutation. We'll be discussing the differences between selections and screens, and the differences in genetic analysis between single-celled and multicellular organisms. Before we talk about the Ames test, let's make sure we understand how wild-type and auxotrophic mutant bacteria grow in the lab. Wild-type bacteria can grow in minimal medium, which supplies the chemical building blocks for synthesis of amino acids, nucleotides, etc. When many cells are required, bacteria are grown in suspension in a liquid culture of minimal media. To analyze the phenotypes and genotypes of individual bacteria, a few hundred bacterial cells are spread on growth medium in agar in a petri plate. At first, the bacteria are invisible, but after dividing for several hours, the descendants of each single bacterium form a clump of cells called a colony. Every cell in a colony has the same genotype. Wild-type bacteria are called prototrophs. All of their biosynthesis pathways are functional, and so they can grow on minimal medium. Auxotrophs are mutant bacteria that cannot make a compound needed for their growth, such as an amino acid. For example, His- auxotrophs cannot make the amino acid histidine because they have a mutation in a gene for one of the enzymes in the histidine biosynthesis pathway. Because they cannot synthesize histidine, His- auxotrophs can grow on minimal medium only if it's supplemented with histidine. The Ames test involves reverse mutation of His- auxotrophs to His+ prototrophs. Here's how it works. His- auxotrophs are grown in minimal medium with a histidine supplement in the presence of a compound that might be a mutagen, or in its absence as a control. Rat liver enzymes are present in both cases. These enzymes are like human liver enzymes that might chemically alter the suspect compound. They are added because we want to know whether the compound is a mutagen when ingested by humans and exposed to liver enzymes. After many cell divisions in the presence of the suspect mutagen, an aliquot of each bacterial culture is plated on minimal medium, where His- auxotrophs cannot form colonies. Only His+ revertants can. A result like the one shown indicates that the suspect compound is indeed a mutagen. The few colonies from the culture grown without the suspect compound are spontaneous revertants. Many more revertants were present in the culture grown with the suspect compound, meaning that this compound increased the mutation frequency. Note that relatively rare His+ revertants in a large population of His- auxotrophs could be detected easily through selection. In a selection, only the organisms you want to count or identify live. The minimal medium selected for His+ revertants. Without a histidine supplement in the minimal medium, most of the cells in the culture, His- auxotrophs, could not form colonies. Only the His+ revertants could. The easy selection for His+ revertants is the reason why the Ames test reverts His- mutants to His+, as opposed to inducing forward His- mutants in a His+ culture. His- mutants induced in a culture of His+ cells would be identified in a screen. In such a screen, wild-type His+ bacteria grow in minimal medium plus histidine to support growth of any His- bacteria that arise. After some time, single colonies are analyzed to determine if they have undergone forward mutation and are His- auxotrophs. Colonies are replica-plated on minimal medium. Colonies that fail to grow without the histidine supplement are His- auxotrophs. You can see that a screen is much more work than a selection. Forward mutants arise much more frequently than reverse mutants do. The reason is target size. Forward mutants, His+ to His-, can be made by changing any one of many different base pairs in many different genes. In contrast, reversion, His- to His+, requires that a specific mutant base pair be changed back to wild type. Nevertheless, the Ames test uses a selection for revertants rather than a screen for forward mutants. Even though the revertants will be very rare, bacteria are easy to grow in large numbers, and selecting for rare mutants is still much less work than screening for relatively frequent ones. Selections for rare events like specific reversions are often not practical in multicellular organisms like fruit flies because so many animals must be grown to find rare revertants, and growing multicellular organisms chromosomes is much more labor-intensive than growing bacteria. The main reason is that organisms like fruit flies pass their genes to their offspring through specific cell types, germline cells that produce eggs or sperm. To detect mutants in the chromosomes of eggs or sperm, you must mate flies and examine their offspring. For this reason, Muller's system for detecting mutations in fruit flies entails a screen for forward mutants. Muller's mutation detection system depended on a genetic tool called a balancer chromosome. Scientists made balancer chromosomes to use in genetic experiments where it's necessary to prevent chromosomes from crossing over during gamete formation. Balancer chromosomes have all the genes a normal chromosome has, but they are in scrambled order. The scrambled gene order prevents normal pairing between a balancer and its wild-type homologue during meiosis, and thus limits crossing over. Crossovers that do occur result in chromosomes so severely malformed that they cannot support viable offspring. Thus, only intact balancer and normal homologues are seen in the progeny of parents with one normal homologue and one balancer chromosome. No recombinants. Muller's experiments involved an X chromosome balancer with a dominant mutation in a gene called BAR. Flies with the BAR mutations have bar-shaped eyes instead of normal round ones. BAR eyes are thus an outward signal that the fly has the balancer X chromosome. Muller asked if X-rays cause mutation. A simple way to answer that question is to determine if an X chromosome exposed to X-rays can be transmitted to a male. The reason is that as males have only one X chromosome, any gene functions destroyed by X-ray-induced mutations are not supplied by another homologue. Also, the mutant phenotype most commonly caused by the absence of a gene function is lethality. The way Muller did his test was to X-ray males and ask if their daughters could give their sons the X-rayed X chromosome or only their maternal X chromosome. The scheme of Muller's experiment is shown here. Muller X-rayed male flies, and while of course the DNA in all their cells was exposed to X-rays, the only DNA that is passed to future generations is the chromosomal DNA in sperm. The X-rayed males were crossed to females that carried a balancer X chromosome, and many F1 females with bar eyes were recovered. These females had one of their father's X's and a balancer X. Note that each F1 female had a different paternal X chromosome from a different sperm. So if X-rays induce mutation, each F1 female has a paternal X chromosome with independently induced mutations. Each individual F1 female was put into a separate food vial and crossed with wild-type males. If a particular female had an X-rayed X chromosome with a mutation that destroyed the function of an essential gene, her sons would all have bar eyes. They'd all have the balancer X. None of her sons could have the X-rayed X chromosome. Females with X-rayed X chromosomes that did not have mutations in essential genes would have non-bar-eyed and bar-eyed sons. Either of their X chromosomes could support viability. What Muller found was that when the P generation males were X-rayed, most of the F1 females had only bar-eyed sons. When the P males were not X-rayed, most of the F1 females had both non-bar-eyed and bar-eyed sons. The conclusion he drew is that X-rays do indeed cause mutations. It is important that you understand the necessity of the balancer X chromosome to this experiment. In the F2, bar eyes served as a marker for males with the X chromosome that was not exposed to X-rays, and non-bar, normal eyes served as a marker for males with the X-rayed X chromosome. For the bar plus and bar minus alleles to serve as markers in this way, no recombined chromosomes could be present in the F2 males. If the F2 males had crossover chromosomes, both bar-eyed and non-bar-eyed males could have X-ray-induced mutations that originated from the F1 female's paternal X chromosome. The balancer prevented crossover chromosomes from appearing in the F2 males, which was crucial to Muller's experimental design.